Hi, I'm Ella Buchanan.
I'm Sage McKinley-Smith.
I'm Maddie Bridge.
I'm Ben Davenport.
I'm Sia Peterson.
I'm Oliver Kendall.
So in this presentation, we're going to go over a brief introduction, and then we'll go over the methods and results of our research. We'll have a quick discussion, and then just talk about the future of our project. So here at CSD, we produce about 10,000 gallons of biodiesel per year. But unfortunately, 10% of that production comes in the form of waste glycerol. One of the issues we have on campus is that we don't have a good way of disposing of or storing this glycerol. So we were investigating in this research project, the first component of our research project, in how we can best dispose of or store this waste glycerol. We started investigating the concept of bioremediation. So bioremediation is a treatment process that uses biological organisms to naturally break down potentially hazardous substances so that they are less toxic or harmless. As you can see here, the way bioremediation works is a fungi, a plant, or a bacteria would consume the contaminant or pollutant, turn it into food, and then produce water and carbon dioxide, which are completely harmless. We investigated fungi, which is mycoremediation. It's remediation of contaminated soil and water or landscapes using fungi. Just a little bit of fungal ecology. Everyone's familiar with the typical mushroom structure, which is the first part of a typical fungus, but the second more important part of the fungus is the mycelium, which is the root structure. And the mycelium is what produces the enzymes that allow the fungus to break down food. It's also what is the key to microremediation because these enzymes also have the ability to break down potentially harmful substances like petroleum, pesticides, or bacteria. Microremediation is typically associated with wood-rotting fungi species because a lot of contamination is a lot of pollutants like petroleum have very similar molecular structures to wood, which is convenient. The second part of our research project concerns food security. Here at the Island School, as you may have heard from the other research project, we are not incredibly food secure. We import a lot of our food. And our non-meat protein sources mainly right now are tofu and beans. So we wanted to use part of our research project to investigate how we could find a more sustainable and more cost-effective protein source. So we started looking at edible mushroom cultivation. For both parts of our research project, we'll be using oyster mushrooms, cultivating them in our lab as a locally sourced and efficient way of producing a lot of non-meat protein sources for the dining hall. Yes. Our research objectives overall are to screen and isolate oil-degrading fungi to be used for remediation, to measure the efficacy of using Phoenix oyster mushroom for the microremediation of soil contaminants, and to investigate the viability of a sustainable mushroom cultivation system here at the Island School.
Okay, so the first part of our methods is the edible mushroom cultivation. So as Ella was just mentioning, as you may have heard from the food security team, we're pretty food insecure here at the Island School. So part of our research project is trying to increase the food security of our non-meat protein sources, which are traditionally tofu and beans. And we would do this by hopefully creating a system to cultivate 50 pounds of mushrooms a week on campus. So mushroom cultivation life cycle starts with inoculation. So this word basically means this is when we mix the spawn with the substrate, and the substrate is what the mycelium will be growing on. So as you can see here. And then we incubate it in an incubation chamber. So this allows the spawn to run until it looks like this. And this stage is complete colonization. This is when the mycelium has spread throughout the entire bag. Once it has reached this stage, the mushroom will begin to pin, as you can see here on the bottom right. These are when tiny mushrooms called primordia will begin to appear, and those primordia will eventually grow into full-size mushrooms and in a fruiting stage. Once the mycelium has obtained enough resources to produce a new flush, it will repeat the pinning and fruiting stage over and over. So the way we apply this on campus is we collect used coffee grounds from around campus, we sterilize them, and then we pack them into bags, as you can see on the right. Then we inoculate those bags and also some containers with the mushroom spawn, and then we incubate it in our incubation chamber. Once it is completely colonized, we move to the fruiting stage, in which we expose the bags to high oxygen, high humidity environment. This causes them to fruit, and we then collect the mushrooms and collect data on them to see how efficient our cultivation methods were. So the next part of our research is fungal cultures. The purpose of this part is to try and maintain a consistent in-house supply of mushroom spawn so that we don't have to buy it online. So we use potato dextrose agar, or PDA, which is a nutrient-rich substrate that helps us maintain cultures. And we grow the mycelium on there, hoping to avoid contamination. As you can see on the left, we sterilize everything. And then the next part after we have cultivated it is fungal adaptation. So in addition to creating those pure cultures, we also attempt to adapt them to be able to consume the contaminants. So we do this by first culturing them, as I just mentioned, and then screening. We place them on a plate of minimal salts medium, or MSM, which the only carbon source on that plate is a 1 to 3% dosage of the contaminants. And so if it is able to grow on that MSM plate and consume those contaminants, we then move to the isolation phase, which is when the mycelium is transferred from the minimal salts medium plate back onto a fresh plate of PDA to allow it to grow, because the PDA is the optimal growing environment for the mycelium. We repeat this process, increasing the dosage each time until we have a strain that is fully adapted to consume the fungi.
The next part of our methods is focused on the microremediation part of our project. For our contaminants, we had biodiesel, motor oil, vegetable oil, and glycerol. This is because these are contaminants that we are producing and or storing on campus. We took soil from the compost pile and we put those into bins, as you can see here, and biotubes, as you can see on the far side of the slide. We then put different concentrations of the contaminants inside these containers, and then we inoculated them with Phoenix oyster mushrooms. Throughout the study, we took different tests on the soil to see how it was affected by the presence of mycelium. One of those tests was the pH. For the pH, we were looking to see how it was affected and how the presence of mycelium altered the acidity or alkalinity of the soil. Another test was the earthworm toxicity test. This is where we had about 48 hours to have the earthworms clear their intestines. And then we put them into petri dishes with some of the soil that was contaminated but has hopefully been remediated. To give you an example of some of the tests that we ran, we looked at their change in the weight. We also designed a scale to interpret the qualitative data, such as light sensitivity and reaction to touch. So here you can see we created a scale of 0 to 4, 0 being unresponsive and 4 being having an immediate active response. We also took observations of their behavior and the behavior of the soil.
So now we're going to look at the results for the microremediation portion of this project. So as Maddie talked about, we looked at the weight change of the worms during before and after the 48-hour period they spent in petri dishes with contaminated soil. The graph on the left shows the soil that was inoculated with pure fungal cultures, while the graph on the right shows the controls that was not inoculated. As you can see, there are some abnormal changes in the weight of the earthworms, particularly with vegetable oil and glycerol, where the worms on average gained weight. This may mean that they're able to consume the contaminants or remediate the soil through that process. However, we need to investigate this more because the earthworms are a foreign species to the Bahamas, and we are not able to introduce them to the environment yet. We also looked at the light sensitivity of the worms after the period they spent in the petri dishes. Like Maddie said, we used the 0 to 4 scale as represented by the x-axis on these graphs. To do this, we simply shined a flashlight on the worms and then observed their response. For glycerol, we observed that 2 of the worms were unresponsive and were likely either dead or unhealthy. But the majority of the worms in the glycerol were healthy and showed a normal response. With motor oil, however, 3 of the worms were either dead or unhealthy, but a few of them showed a normal response, which is good. We also looked at the touch sensitivity of the earthworms. To this, we simply touched the worms and again observed the responses. With glycerol, 2 of them were again either dead or were showing unhealthy signs, but the majority of them were healthy. With motor oil, 4 of them were not looking so good. They were they were either dead or, again, very unhealthy, and a few of them showed a normal response. We also looked at the pH of the soil that was contaminated as well as inoculated. Here's the graph for glycerol, where the y-axis shows the actual pH of the soil. The dotted green line that you see is the pH of only glycerol, only the contaminant. The general trend of the soil is that the soil becomes less alkaline, which may be a good sign because it is returning to a more normal pH for the soil here. And then here's the graph for motor oil, where you see a similar trend of the soil becoming less alkaline. We also measured the linear growth of the pure fungal cultures in the MSM plates as well as the MSM plates, or the PDA plates as well as the MSM plates. The PDA is used as somewhat of a baseline because it is an ideal growth condition for the fungi. It grows at, or it grew at a 5.6 millimeters per day on average. This is a normal healthy growth rate for the fungi. And looking at the MSM growth rates, they grew either around that rate or at a faster rate, which is a very good sign. It shows that the fungi can consume the contaminants and grow from that.
Okay, so next we looked at results for edible mushroom cultivation. The first thing we looked at was the cost analysis. So we did this by, we're able to calculate how much it would cost per pound to cultivate mushrooms here on campus, and we did this by using a program called Grow Cycle, and we compared that to how much we are paying now for our imported other non-meat protein sources such as mushrooms and soy products, where we got the data from the food security team. As you can see, it costs about 40 cents cheaper to grow our own mushrooms here than to import non-meat protein sources. This not only makes our campus more food secure, it also makes our campus more sustainable because soy products require a lot of resources to grow. Next, we looked at species to cultivate, and out of the 4 species we investigated, we found that the summer white oyster mushrooms were the most promising. These mushrooms are able to thrive in our tropical climate. They also produce a high yield, which makes them optimal for growing and serving in the dining hall. And they're able to be cultivated on a wide range of substrates, which are all available to us here on campus. The last thing we looked at was contamination. And as you can see, this picture indicates what we have identified as trichoderma mold, which is a prolific contaminant here. We have about a 38% contamination rate, meaning that if we were to scale up our mushroom production to be able to produce those 50 pounds of mushrooms per week, we would need to re-examine our sterilization protocol.
All right, now for the discussion. The first part of the discussion is the fungal adaptation. And the example on the left is a Phoenix oyster mushroom on MSM and 3% biodiesel going through the screening process. And the sample on the right is on a PDA sample at 3% biodiesel, and that's the isolated strain. And so for the fungal adaptation, it was successful, but in order to start using it to remediate the soil, we're going to need to keep screening it to get up to around 10% contamination of each of the contaminants. Then for the earthworm toxicity test, it made us want to reevaluate our soil remediation priority, putting motor oil at the top because it is the most toxic, and so we'll need to investigate that in the boneyard, or in the waste disposal site on campus, and clean that up first. And then glycerol was the second priority, and that was due to the earthworms eating it and being able to clean it up successfully. And then the other contaminants are below it because the biodiesel is being used on campus, and then vegetable oil is a natural product that does not harm the environment. And then for the edible mushrooms, we grew the mushrooms successfully, but the only problem we have with scaling up our production to 50 pounds of mushrooms is the amount of substrate that we need to collect, with the monthly requirement being around 600 pounds of substrate that we need to somehow source from importations from Nassau or around other islands. And then we get around 30 to 60 pounds of substrate from on campus from the coffee collection. And so for the future of the project, first we're going to have to source some capital funding, and then we're going to expand production to hopefully a shipping container so we can grow the 50 pounds of mushrooms as well as monitor and control the environment a lot easier. And then the third part is to begin using the remediation in the waste disposal site here on campus. And so in conclusion, to revisit our objectives, we successfully did in vitro oil degrading fungi and isolated the strain of that, but we're going to keep keep screening and isolating until we get to the right concentration. And then we're also going to start using it to monitor the soil after we do that. And then we also successfully cultivated mushrooms on campus and completed that objective. So here's our literature cited. And for our acknowledgements, I'd like to thank our advisors, Dorlin Curtis and Alex Cook. And special thanks to the kitchen team, the comms team, the facilities team, Cam, Mike Cortina, Pam and Chris Maxey, Scott Roberts, and DePer Coffee Shop. Any questions? Yes.
I wondered if you are at all worried about the mushrooms taking up contaminants from the substrates and the extent to which the mushrooms might be contaminated as a food source. If that's something you have to worry about.
The mushroom cultivation is separate from the remediation of the soil, and so the cultivated mushrooms would be on the clean substrate, and the mushrooms that we would grow to remediate would not be used for consumption. Yes, we are using those to see if they'll remediate the soil. Scott.
How feasible is it to use another substrate other than coffee?
He asked how feasible it was to use other substrate instead of coffee grounds. And other substrates do work really well, but for on campus, it was easier to source the coffee just because it was very easily accessible. But you can also use like wood chips or mulch or wood shavings as waste.